It's a real pleasure to be here. Let me pull up my slides. Okay. And here they are. Okay. So as Ishwar said, I am a program director for the BD2K programs, and I'm joined by a few of my colleagues here from NIH, and so I hope that you will get to make our acquaintance as the day progresses. So my goal in this introductory talk is to wow you with the impressive accomplishments of this program, and also to engage you so that you will apply your talents and your abilities to problems that are of interest to NIH. The talk is divided roughly into two parts, and the first part is all about BD2K, and the second part is going to be about the priorities of NIH for FY18. If I don't make it through the whole entire slide deck, no worries. The slides will be made available to you because there's a lot of initiatives that are going on that I think would be of interest to you to look at, and so you will have all that information. If you don't get it here, you will be able to download it later. So no talk about BD2K is complete without acknowledging the hard work and the dedication of Dr. Phil Bourne, who is the Stevenson Chair of Science and the Director of Data Science at the Institute of Data Science at UVA. BD2K was launched in 2013, and it is and will continue to be a trans-NIH initiative to address the challenges and opportunities posed by biomedical big data. And you can see the QVR code to learn more about BD2K. As the first Associate Director of Data Science at NIH, Phil brought to our institutes the concepts of the FAIR principles. That is, that data and digital objects should be findable, they should be accessible and interoperable and reusable. NIH has embraced this concept wholeheartedly, and today we will see the accomplishments of many of the BD2K programs which have taken roots and now flower in all of our NIH programs. So in the interest of time, I can't possibly tell you all of the wonderful highlights that have happened in this program, so I'm just going to hit a few of them in the areas of indexing and software development and training in our centers of excellence. You know, in order to enable data and digital objects to be findable, you have to be able to actually find them. And so we supported the BioCaddy team to develop novel and innovative indexing strategies that would help researchers find data they wouldn't necessarily have access to. It's sort of easy to find the data you know about, but what's really hard is to find the data that you don't know about. And so that was really the mission and the goal of BioCaddy. And they did so by developing innovative indexing strategies to find data that required a fairly large community effort to develop the metadata specifications, because without metadata, data is really pretty meaningless. And they supported a crack team of software developers to implement new and interesting ways to search data through machine learning algorithms. The prototype that they developed, and you can access it by going to the website biocaddy.org, is called DataMed, and it indexes over 66 very different data repositories. That's 1.3 million data sets covering 15 different data types. And the strength of this work is really that they integrated indexing strategies across different databases as a means to make data truly findable. But in order for data to be findable and to adhere to the FAIR principles, there's a lot that really needs to happen on the back end that we as biomedical researchers may not necessarily see. And through supporting different software development programs in data compression, in data provenance and wrangling, in data privacy, and engaging the community in data science through crowdsourcing initiatives, we're able to accomplish a lot of back-end goals. And I was particularly excited yesterday about Madeleine Bell's concept of open data and engaging data science through community access and participation, and in a way making data science a two-way conversation and not a one-way conversation. And I think that's a wonderful way to go in the future. And to this end, I want to highlight just one exciting outcome of our Data, Software, and Tools initiative, and that's the work by Dr. Wu and Dr. Su that were able to harvest and import data from many different sources that included over 15,000 species, and in such a way that the data was structured so that the community could explore this data on their own. And the work is called myvariant.info and mygene.info. And so I encourage you to explore these two new and innovative ways to engage the community to understand genes and variants. A core goal of the BD2K program is to invest in a new generation and to bring data science skills into our biomedical science research programs. So with over 87 grantees training well over 100 graduate students, postdoctoral fellows, and clinicians, we're able to accomplish this goal to improve big data skills in our biomedical scientists. You can learn a little bit more about our training programs by going to the Training Coordination Center. And one exciting program that I want to highlight for you is called the Data Science Rotation or Roadmap program. And it's really a partnership where we can support an investigator who's really in the first few years of their independence career to partner with a senior data scientist so that they can take their research into new directions. And that's exactly what Andre Iremia did with a new collaboration to apply machine learning in the analysis of his neuroimages. And from that work, he was able to form 2 new collaborations with engineers and data scientists. And I think that that's an exciting area, and it's wonderful for NIH to see new investigators really engage in data science. Finally, I want to highlight just a few of the wonderful things that have happened in our BD2 Centers of Excellence. We have 13 centers with over 640 publications and over 120 different tools. It would be hard to pick just one highlight, and this session that's going to follow will give you a sample of some of the accomplishments, but you can read about a number of these highlights in a special issue of Biomedical Computation that was just published, and so I encourage you to do so. So in this crazy subway diagram, this lets you see visually all the interconnected work in the areas of data science that the centers contributed to. And I would argue that this interconnection and collaboration would never have been possible if we had just funded each of these investigators individually by separate R01 programs. And it would never have been possible if we didn't coordinate this effort with our wonderful coordination center, which is led by Dr. Pei-Pei Ping. Through these collaborative and coordinated efforts, we were able to accomplish far more than we could have ever done as a single investigator program. And like the metro in Prague, it's all accessible by a simple push of the button. So what's next in BD2K? As we move from phase 1 to phase 2, where we've piloted different ideas in the FAIR principles, and now it's really time to stand up the FAIR principles and make data usable and make it reusable. And that's just what we're going to do with our Data Commons pilot phase, where we will develop methods to support the global unique identifiers for data and digital objects, where we will create workspaces for computations and develop new cloud-agnostic platforms that will integrate 2 different cloud platforms together for the commons. We'll, of course, continue our work in developing APIs and API standards and new approaches to data access, data ethics, data privacy, and data security. We anticipate that within the first 18 months after October of this coming year, we will have stood up a Commons pilot with the 3 targeted data sets of TopMed, GTEx, and some of the model organism databases. So in the remaining time, I just want to give you a whirlwind tour of what our priorities are for FY18 as set out by our director, Dr. Francis Collins, and they are enhancing new technologies to support fundamental science, to develop new treatments and cures for diseases, and promote health and enhance stewardship of our biomedical workforce and our biomedical research. I'm sure that you've heard of the BRAIN initiative launched in 2013, which will produce a clear and dynamic model of the brain that can show for the first time how individual cells and complex neural circuitry interact both in time and in space. But what I want to highlight for you is a new breakthrough that's happened really just in May of this past year. And it includes creating a three-dimensional model of two different spheres of the brain, and from this model, understanding how neurons can migrate from one sphere to the other. And what's really interesting is that the researchers, Dr. Pasca and his collaborators, use this model to understand how neurons from patients who have Timothy syndrome are different from normal neuron behavior. And in this way, they're hoping that this can lead to new treatments for this debilitating autosomal rare disease. There are a number of initiatives that are coming forward this year in FY17 and 18 for the BRAIN initiative, include developing experimental standards for new experimental protocols, to develop and support a data archives for all the immense amount of data that's going to be a result of the BRAIN initiative, and to develop new analysis and visualization tools that can leverage this data from the BRAIN initiative to help develop new models. So if you are interested, do please contact Dr. Greg Farber. Other priorities in FY18 include developing and supporting cutting-edge technologies to advance treatment for sickle cell disease, and focusing on cancer immunotherapy to discover new immune targets, and as well as intervening in the opioid epidemic. I'm pretty sure that you've also heard of the All of Us research program that's standing up a cohort of more than 1 million volunteer participant patients who will link their genomics and other patient omics data with their healthcare records to provide a foundational, fairly large data set that will enable researchers to learn from this data to prevent diseases and develop new therapeutics and new drugs. What I want to tell you is one highlight from this program, and this did happen about a year ago, and it's the epigenomic analysis of genes in rheumatoid arthritis in both the knee and the hip. And what these researchers found was that the methylation pathways for knee and hip is very different. And so what's hoped is that because these are different, we can now develop targeted therapeutics rather than the cocktail of drugs that we now prescribe for rheumatoid arthritis. There are other initiatives that do support computational biology and mathematics. As Ishwar said, we do have our BISTI, Biomedical Information Science and Technology Initiative, as well as our Interagency Modeling and Analysis Group that looks at multiscale modeling from cells to behavior, and our NSF-NIH Joint Program in Mathematics, which brings novel and new mathematics to the biomedical research area. It doesn't necessarily have to be new mathematics for NIH, but we do hope to engage mathematicians and statisticians to come to NIH and to apply their skills to biomedical research. So you can find out more about BISTI and these other initiatives by going to the webpage. And now I'm just going to highlight a few initiatives from different ICs across NIH that you may be of interest to you. The first one is discovering novel gene targets that really looks for folks to identify, validate, and or functionally characterize genetic variations and haplotypes that might be associated with addiction. This is Jonathan Polak's initiative, so do please contact him. There's a new initiative to develop new and computational models to help identify drugs or drug combinations that might be a potential efficacy in Alzheimer's-related treatments. So I think that might be of interest, and Susanna would certainly be a contact. Something that I'm pretty excited about, because it sort of borders on citizen science, is to develop new information and new data science approaches to help gather, manage, and present health-related data to patients and people. So this is really to develop apps that will help people understand their own personal health. We have an initiative to develop convergent neuroscience approaches so that we can establish causal or probabilistic linkages and models that will explain psychopathology. And what's really interesting about this particular initiative is that the FOA must consist of at least 2 different collaborative sites that are essential to provide this research moving forward. And that's something that you're going to see in FY18 from NIH, is a number of initiatives that do highlight and promote collaborative team science-based research. One will be coming from my own IC. So you might be surprised to realize just how much NIH spends, both in our extramural and our intramural program, in areas of computing, of systems infrastructure, and software development, and education and workforce, just in data science and computational sciences, informatics. It's about $1.4 billion last year, which is a significant amount of money, but it's spread and diffuse across all of the ICs. So it's really important to tap into the many different ways that you can support your research in these areas. And finally, I think it's always important to look at the statistics of our success rates, which I do believe for the first time in many, many years have hit an all-time record of totally about 19% success rate, and in different ICs with different priorities have different success rates. I'll just take a minute here, okay? And finally, Dakuyu, thank you so very much for the time, and I hope that the information was valuable and insightful, and I think that it's, thanks. And I don't know if there's time for questions, so. Oh, okay. Okay, so there's time, all right.